Hi, I'm Russell Tate, CEO and founder of Polyactiva, speaking to you from halfway around the world in Melbourne, Australia. This is our first presentation at OIS, and it is a pleasure to have the opportunity to introduce you to our company. We have a truly unique drug delivery technology that we've used to develop ocular implants that substitute for existing drop therapy. Our first two assets are in glaucoma and post-surgical care. With the recent demonstration of our technology is safe in human eyes, we're now looking to expand our product pipeline. I'm excited and hope you enjoy the presentation. Polyactiva is a privately held clinical stage ophthalmology company based in Melbourne, Australia. We have two assets in clinical development, an ocular implant for the treatment of glaucoma and an ocular implant to provide antibiotic prophylaxis following cataract surgery. Both implants deliver known drugs, so can be registered through a 505(b)(2) regulatory path. We own each asset outright and they are protected by patents with composition of matter claims. We have an outstanding clinical advisory panel that oversees each of our clinical programs. We use a common delivery technology that provides separate control over drug release and implant biodegradation to ensure each product is tailored for its market application. We have single agent clinical assets in each of the glaucoma and post-surgical markets and are seeking to extend our pipeline in each of those markets with multiple agent implants. We are targeting a 6-month treatment period in the glaucoma segment and to substitute existing standard of care drop therapy with a single implant in the post-surgical segment. We also intend to expand our portfolio to include back of the eye assets to treat conditions such as wet AMD and DME. Polyactiva has developed a truly unique and differentiated drug delivery technology. The technology does not involve blending of drug through PLGA or any other polymer matrix. Rather, it involves use of polymeric prodrugs, where a drug substance is chemically linked to a polymer backbone, and the chemistry of that linkage used to control drug release. The implant is produced by first synthesizing a drug incorporating monomer, which is then combined with comonomers to build a custom polymeric prodrug. This in turn is transformed into an implant suitable for syringe administration. For front of the eye application, the implant is administered in the ophthalmologist's office to the inferior angle of the anterior chamber by clear corneal injection. Immediately after administration, the implant takes up water from surrounding tissue to trigger drug release. A constant daily dose of drug is then delivered for the desired treatment period. A cross-linked polymer network ensures implant integrity during the release period, after which the implant biodegrades and leaves no residue. The technology is tunable. Exquisite zero water release is achieved over the treatment period, which can be as short as 1 week or as long as 2 years. We can control the rate of biodegradation, but also when it starts. With our implant, the biodegradation process kicks in at about 26 weeks after administration and is complete 6 weeks after it starts. There is no residue and the degradation byproducts are non-toxic. More than 90% of existing drugs can be delivered with our technology. Provided the drug can be produced synthetically, we can deliver it. We've used our technology to build a glaucoma implant that releases latanoprost-free acid, the active agent of latanoprost. We built 2 forms of the implant from a common chemistry platform. The 2 forms have different biodegradation profiles to ensure we achieve the target 6-month treatment period. We studied both forms of the implant in a validated animal model and assessed them weekly. Both implants were found to be safe and well tolerated. In both cases, an IOP lowering effect equivalent to Xalatan drops was observed during the treatment period. In dogs, one form of the implant was found to biodegrade at about 20 weeks and the other at about 30 weeks. We expected the 20-week implant would probably translate to a 30-week implant in human subjects, so selected this for initial clinical development. We then progressed with a phase 1 first-in-man clinical safety study conducted here in Australia. The study found the implant to be well tolerated in the subjects recruited. The biodegradation period was confirmed to be between 24 to 36 weeks across the population, and importantly, there was no sign of any implant movement. While the study was not designed to show efficacy, we did see evidence of an IOP lowering effect. Topical PG analog therapy was ceased when the implant was dosed and not reinstated until IOP returned to a normal clinical care range, as illustrated by the plot shown to the right of this slide. We've now taken the same implant into a phase 2A dose ranging study. We are studying 3 doses in a single ascending design and will recruit up to 10 patients per dose cohort. The primary efficacy endpoint is mean diurnal IOP at 12 weeks compared to baseline IOP, but we'll also study the mean diurnal IOP at 26 weeks. We hope to report interim findings for this study early to middle of next year. Our second clinical asset is an ocular implant to substitute for peri and postoperative antibiotic therapy following cataract surgery. The implant delivers levofloxacin, a fluoroquinolone antibiotic. Levofloxacin is delivered with a biphasic release profile. First, a bolus dose is released within 15 minutes of administration. This substitutes for the bolus dose of fluoroquinolone typically administered by surgeons during cataract surgery. The bolus dose is then followed by release of a constant daily dose of levofloxacin for about 5 to 7 days. This substitutes for topical antibiotic therapy after cataract surgery. We've initiated a first-in-man clinical safety study with this product and again hope to report interim findings for this study early to middle of next year. Now that we've moved our first 2 assets into the clinic, R&D resources have been freed up for new product development. We have directed those resources to development of new products and extension of existing. For example, we are developing a levofloxacin steroid combination implant that would be an obvious life cycle extension for the levofloxacin alone product. We also intend to extend our glaucoma franchise with additional single agent implants and a combination implant of 2 agents. Finally, we've started development of a back of the eye implant to treat wet AMD, DME and/or RVO. So, in conclusion and to recap, we have a unique and differentiated platform technology that allows us to separately tailor drug release and implant biodegradation to meet the needs of the intended application. The technology is applicable to a wide range of drugs used in ophthalmology. For each drug class we've studied, we've filed patents to protect the relevant asset with composition of matter claims. We have 2 assets in clinical development in 2 of the largest markets in ophthalmology. And finally, we're seeking to develop new products with our technology to create a pipeline of assets. I trust you enjoyed the presentation and thank you all for your interest.